Okay, hello, hello. Are you audible right now? Someone in the chat, can you tell us? Are you audible right now? If we are, just let us know, please. Okay, hello, hello. Are you audible right now?
Hello.
Hello, yeah. Mohammed, I'm so
Yes,
sorry. We weren't
this
really
is fine.
audible when we started, so we'll have to start again.
This is fine.
All right. Uh, m- I think we're audible now. Thank you so much, Mayur, for letting us know. All right. So, Mayur, sorry, Mohammed, can you just take us through this again, the introduction to microbes?
Yeah, basically, microbes are organisms that cannot be seen without a microscope. They are both unicellular and multicellular. They are the earliest form of known forms of life. They include bacteria, fungi, and protists. They are both useful as well as harmful. As I set an example in the small intestine, we have useful bacteria, uh, and in causing diseases also there is harmful bacteria which destroys the cell from the inside and cause of us many symptoms and painful diseases.
Absolutely. So that is a small introduction to microbes. Microorganisms, as they're also called, is like they're very small organisms, and that is why they cannot be seen without a microscope. So a living organism which is very small in the eye and that cannot be visible to the naked eye is essentially a microbe or a microorganism. Okay, so these are the three types of microorganisms that we had, further broken down them into. So bacterias, fungi, and protists. So bacterias, as we know, belong to the Monera kingdom. Protists and fungi have their own, kingdoms when we classify living organisms. So bacterias and protists, they are usually more unicellular, but mushrooms and fungi are usually more multicellular. Although protists can also be multicellular, and their shape and their structures can also vary from organism to organism, right? So the bacteria cell structure is like quite generalized and quite fixed, but for protists and fungi, it can vary from organism to organism. But Mohammed, can you take us through a overview of how these cell structures can look?
Yeah, so basically bacteria are prokaryotes, which mean they are single-celled organism and they have very simple uh, cell structure. They don't have organ or small organs inside the cell like fungi or protists have. Like for example, fungi has a chloroplast, but bacteria doesn't. They have also mitochondria, but bacteria doesn't. The bacteria also lack cell DNA wall, basically, and they have a small extensions on them, like the flagellum and the pili, which help in movement and detection of the environment. And they have cytoplasm ribosomes, which are less developed than that of the fungi and the protistid. And they have a capsule and cell wall to protect them from external hazards in their environment. They also have cytoplasmic membrane. Meanwhile, fungi has a more developed cell structure. It has a chloroplast, nucleus, nucleolus, flagellum, eye spot, photoreceptors, contractile vacuole, which mean it can use light as a source of energy for its cell functions. And when it comes to protists, it can be unicellular or multicellular like mushrooms. And it has scales, cap, um, mycelium, which help it in absorbing nutrients from the soil or dead organisms. And all these three uh, kingdoms feed on other organisms, except for some species of the fungi which make its own energy by using light energy,
Absolutely.
which is
So
also called autotrophism. Yeah.
So they can be autotrophs or they can be heterotrophs. So heterotrophs means that they feed on other organisms, and autotrophs mean that they can make their own food, they can make their own nutrients that they want. So when they can photosynthesize, some bacteria, some forms of protists, some forms of fungi, they can photosynthesize and make food for themselves, but other organisms need to feed on the you know as Mohammed said, dead cells or other living organisms as well. Okay, now let's just jump straight into the different specific species of organisms that are useful to us and that are harmful to us, as the name suggests. So, the first bacteria or the first microbe that we will be looking at is lactobacilli. Mohammed, can you give us an overview about lactobacilli?
Yeah, so basically it is a microbe in the small intestine. They are small and have a rectangular shape. They are anaerobic, which means they don't need air to respire. Oxygen, basic, sorry, not air. They do not need oxygen. And the first name can give us a clue about what they do. They digest milk, I think, because it has lacto in its name. So they help in the digestion of milk uh, and transform it into small sugars like glucose and galactose and make it easier to digest.
एब्सोल्युटली ओके एंड जो भी वंडर कर रहा है दोस्तों कि आज हम ज़्यादा इंग्लिश का क्यों इस्तेमाल कर रहे हैं, बिकॉज मोहम्मद इज़ लाइक काइंड ऑफ अ जीनियस एंड he has his roots outside of India, but we specially invited him to give you that global perspective. आपको दुनिया में अलग अलग जगह से हम गेस्ट्स को बुला रहे हैं और आपको एक कॉम्प्रिहेंसिव पर्सपेक्टिव देने की कोशिश कर रहे हैं. सो इसलिए आज ज़्यादा इंग्लिश का प्रयोग होगा, बट डोंट वरी गाइज, इफ़ यू डोंट अंडरस्टैंड एनीथिंग चैट बॉक्स में आप प्लीज़ लिख दीजिए एंड आई विल ट्रांसलेट दैट फॉर यू. ओके मोहम्मद, लेट्स मूव फॉरवर्ड
Yeah, sure.
with the structure of the lactobacilli. So the
Yeah,
image on the
so
right is the image under the microscope, and the image on the left is its schematic diagram. Yeah, take us through it.
So basically, Lactobacilli is small bacteria which can be seen by the light microscope. It is not that too small that we cannot see it, or we need the electron microscope to see it. It has also a simple structure as all of the bacterias. DNA with no cell wall which protected, ribosomes which are less developed than that of eukaryotes, cytoplasm, cell membrane, cell wall, and capsule to protect it from the external environment.
And we can also see that the lactobacilli has like a rod shape, right? That is going to be important when we study it further. But that is how a lactobacilli looks like. And as Mohammed explained the cell organelles of lactobacilli to you. Okay, but now let's just delve into how lactobacilli is used, right? We all know, we all have seen our parents make curd at home. हम मम्मी पापा को देखते हैं दही जमाते हैं वो लोग घर पर. तो वो कैसे जमता है, the process of making yogurt. Mohammed, can you take us through the science of how it works?
So basically, yogurt is made from milk with the help of bacteria which ferment the milk and transfer it into lactic acid and which lower the pH and make yogurt by adding basically water and milk and lactobacillus. Lactic acid is formed which help in the fermentation of the milk and make transferring it into yogurt.
Absolutely. So we can see that there is this lactose sugar, right? So whenever there is sugar, what does an organism do? It respires or it breaks that sugar down, right? So the yeast, sorry, not the yeast, sorry, the bacteria will break the sugar down, in a form of anaerobic respiration. But Mohammed, can you tell us like why lactic acid is formed and why not our aerobic oxygen and water?
uh, the bacteria is anaerobic, which
Absolutely.
mean it will have to use lactose because without oxygen, it will not be able to reuse the glucose. So lactose is formed as a way to which is lactose is reusable. The cell can remake lactose, unlike glucose.
Absolutely. So the cell will remake the lactose, and the lactose will not be broken down completely because there is not, it is anaerobic respiration. That means there is not total respiration that's taking place because oxygen is not present. So there's partial, breaking down of the lactose molecule, and that is why lactic acid will be formed, right? So that is what is going to happen in the bacterial mitochondria, where lactose will be broken down into lactic acid. Now, after lactic acid is formed, how does that lactic acid, just convert into yogurt or curd?
Hello.
Yeah, yeah, you're audible.
Yeah. So basically, the coagulation is a process of forming clots. In this case, milk clot, because the bacteria ferment the milk, and it gets separated from other substances. And when the process is complete, the yogurt is formed.
Absolutely. So, guys, you might have noticed that when there's milk, milk is like liquid, but you can see through, like it's like kind of semi-transparent. But when you add bacteria, when you add, like when you ferment the curd, when you जब दही जमाते हो आप, after some time you can see that the solid separates from the liquid. Like the solid just kind of separates from the liquid way, and that is the process of coagulation. So coagulation मतलब basically जो हल्का हल्का इकट्ठे जो वाइट वाइट पार्ट है उसका, जो प्रोटीन्स है, मिल्क प्रोटीन्स, वो इकट्ठे हो रहे हैं और वो एक साथ जुड़कर एक एक लंप बना रहे हैं, एक क्लॉट बना रहे हैं, दे फॉर्मिंग अ क्लॉट टुगेदर, एंड दैट इज द प्रोसेस ऑफ कोएगुलेशन, राइट? हमने कोएगुलेशन पढ़ा है सीवेज ट्रीटमेंट में भी, वेयर all the little particles that are there, or that are the impurities, they are coagulated, they are just kind of, एक अलग अलग जगह से they are taken, they are dissolved in the liquid, and then they form a lump together, they form a coagulate, they form a clot together. So that is how the solid just kind of separates from the liquid, and now the liquid is more transparent, it's more watery, and the solid just now contains all the proteins and all of the other uh, nutrients that we want in the yogurt. So that is what is happening. So the lactic acid is just basically lowering the pH, and this pH, when the pH of the curd lowers, because it is an acid, right? The lactic acid is ultimately an acid, it's a weak acid, and it will lower the pH to about 5 to 6, and that will cause, like, the proteins, because we know that the enzymes in the proteins are very specific to the pH, right? So then the coagulation process will be triggered and the clots will be formed. Mohammed, do you have anything to add to this?
No, actually, no. It is a simple process. Like it is a fermentation, and milk is separated from the other molecules. Yogurt is formed. It is in outcome is yogurt, and basically, what is the whole this whole process is only fermentation.
Absolutely. So that is what is happening for the yogurt making process, right? Now, uh, one more thing, one more advantage that is there because of this bacteria, that is involved in the yogurt making process, is that the acidic pH actually kills all the harmful microbes in the yogurt. So we know that enzymes are denatured when the pH changes. We know that enzymes are very specific and receptive to the pH. So if there is a change in the pH of the solution of the environment, then the enzymes in the harmful bacteria, for example, you know if you or me, we are put into a tub of acid, right? Then we will start, our enzymes in our cells will start to denature, and we will start to kind of die. So that's what just happens to the microbes, where its enzymes in the cells denature, and it cannot carry out its life processes that we have studied in the chapter in grade 9. We'll also be studying respiration in grade 10. So when it cannot respire, it cannot produce energy, it cannot take carry out all of its life processes, and ultimately it will die, right? So that is what happens to the harmful microbes, and it makes it more, uh, I could say, hygienic for us to consume. Not hygienic technically, but yeah, disease free. So that is what happens due to the lactic acid in the yogurt. Okay, let's move forward. Now, what are the other uses of lactobacilli in the industry? Okay, so Mohammed, can you just quickly take us through the different uses?
Yeah, so obviously you don't need to remember everything in this. It is just to give an idea of the uses of lactic acid in bacteria. So not only in food, but sometimes it is used in medicine to separate some substances. If we have a specific thing that is useful in milk, we use it in medicine. We use it for studies in chemistry. We use it in probiotics. You know that yogurt is very helpful for the small intestine. It has many benefits, which mean that if someone has a specific illness, he can drink yogurt or eat it to make it useful, to make it his usefulness for the small intestine useful. And basically it is a starter culture, which means the water that we extract from the yogurt that is left can be used in dairy foods and non-dairy foods, for example, to make butter and other products which are made from milk. And also there is antimicrobial agents, as you mentioned, that it kills bacteria because it lowered the pH, so it will kill some bacteria, which means it can be used to preserve food or preserve medicines, and it is low calorie sweeteners. It doesn't have a lot of glucose in it. It has specific enzymes. The bacteria has specific enzymes to digest the lactose and transfer it into glucose and galactose. And it has vitamins which are essential for the body, which are needed, especially if you are growing up as a child.
Well, absolutely. So, Momo just gave us a lot of scientific insight over there, and I'm struggling to keep up with this pace there. But there are different uses of this lactobacilli or the lactic acid, that has been formed from this yogurt making a fermentation process. As Mohammed said, industrial uses making different products in biopreservatives in foods. So जैसे हम दही जमाते हैं वैसे हम अलग अलग प्रकार के है ना खाना बना सकते हैं, दूध दही बटर जैसे मोहम्मद ने कहा। वो सब चीज़ें के लिए भी हम जमाने के लिए बनाने के लिए ये लैक्टिक एसिड हम यूज़ कर सकते हैं। And different nutrients, because lactose is ultimately a disaccharide sugar. So if we polymerize the lactose, we can make polysaccharides. And अल्टीमेटली जो लैक्टोबैसिलस जो बैक्टीरिया है उससे हम उसके एंजाइम्स डिराइव कर सकते हैं, उससे उसके एंजाइम्स लेके हम आर्टिफिशियली लैक्टिक एसिड बना सकते हैं। वी कैन कैरी आउट आर्टिफिशियल फर्मेंटेशन यूजिंग एंजाइम्स फ्रॉम द लैक्टोबैसिलाई एंड लॉट ऑफ डिफरेंट यूजेस ऑफ दिस lactobacilli bacteria.
So basically, it has the enzyme lactase, which can be separated from the bacteria and can be used if isolated in many to digest many substances or break down, to be specific, many substances. So the lactase enzyme is extracted from this lactobacilli and can be used in many things like food and medicine and all the chemical industrial things.
Absolutely. So these are like various different uses that you can write about, you can read more about, which is quite exciting, to be honest. And for people like Mohammed who have a passion for biology, I think they really love all these insights. Okay, so again, these are some more uses of lactobacilli that are there in your syllabus, but I think we have covered a wide range of uses, so we can probably just read through it and not dive very deep into this, right? So we spoke about the production of dairy products, so like a starter culture. So that's the use of lactobacilli over there. It is also used in the production of, say, coca, cider. So all non-dairy products, it is used in the production of those as well. Then it mentions about general digestion problems, as Mohammed said. Yogurt can serve as a probiotic and that is really helpful for improving the small intestine. It just kills all the bad bacteria in the small intestine, and it promotes good bacteria in the small intestine. So all the digestion problems over there, some of them can be cured by using yogurt that is made from lactobacilli. So these are some of the uses, and we discussed some of them more in the previous slide as well. Okay, so all these processes over here, all of them follow fermentation. So जहाँ पर भी fermentation हो रहा है, lactobacilli हम use कर सकते हैं. Okay, let's move forward. Okay, next microbe, Mohammed, rhizobium.
So
Yeah, okay, let's just skip to the structure of rhizobium, and probably you can take us through the structure.
yeah, so ribosium is another bacteria.
Yeah.
It has basically this is just a second. I cannot see clearly.
Okay, I can probably just help you with the diagram and you can explain that. So the rhizobium over here, we can see some cortex cells here, and there is the bacteroid over here, and there is some mitosis that's taking place over here. Uh, but I think generally it's almost like the same structure over here, where we can see threads out like nodules and flagella over here, and the bacteroids over here are just reproducing. So it's kind of the same bacterial structure that we have seen for the general bacteria. So it's like they infiltrate into the into the soil and affect the plant nodules. So can you take us through how a rhizobium bacteria can really affect a leguminous plant, or that is or the host that we call?
So through nitrogen fixation, obviously, so rhizobia is bacteria which are in soil. They help plants by uh, through nitrogen fixation by taking the nitrogen from the atmosphere and the soil and make it into smaller nitrogenous compounds which are usable by the plants and help in the process of growing, making leaves, making the fruits of the plant, like you, as you mentioned, the legumes, uh, and many other processes that the plant require. The plant is a host for these bacteria. It is beneficial for the farmers also. If we talk about the financial side of these bacteria, it helps them to reduce the use of fertilizers as it will do nitrogen fixation, basically, which is the transformation of nitrogenous compound which are present into the atmosphere and the soil, which cannot be used by the by the plant into usable compounds which can be absorbed into the plant and be used to develop and grow the plant.
Absolutely. So दोस्तों नाइट्रोजन फिक्सेशन बेसिकली एक प्रोसेस है जिसमें राइजोबियम है ना, हमने पढ़ा है एवरी लिविंग ऑर्गेनिज्म हैज़ एंजाइम्स. सो जो राइजोबियम है उसके पास एक स्पेशल शक्ति है, उसके पास इसका एंजाइम है कि जो हवा में नाइट्रोजन है उसको वो अमोनियम कंपाउंड या नाइट्रेट आयंस में कन्वर्ट कर सकती है. नाउ मोहम्मद, कैन यू टेल अस व्हाई nitrogen fixation is important? Because we know that plants need nitrogen for growth, and that's like that is like important for their development. But why can't they just use nitrogen just like that? It's like they can take it through the stomata, right?
Now, nitrogen in itself is not very useful for the plant because, like, it is a very unreactive compound, so it has to be transferred through processes which the rhizobium bacteria does that for the plant and turn it into a useful compound by combining it with other ions which are present in the soil and giving it ready for the plant to be used through the biological processes which help the plant to grow and develop.
Oh wow. So what that means, guys, as I think he's trying to say कि nitrogen, देखो हमें पता है कि triple bond होता है. There is this triple bond between nitrogen. So that means that nitrogen, the plant really cannot react nitrogen and use it in the way it wants to, right? Because only pure nitrogen is of no use to the plant. So the plant obviously has to make a compound, a biomolecule with the nitrogen that it acquires. So that means that it has to convert it into a form where the nitrogen will now react with other biomolecules, it will react with other chemicals like carbon and hydrogen, and it will form compounds. So just pure nitrogen won't react with a carbon or a hydrocarbon or an organic compound, but a nitrate ion, that is an NO3 minus ion, or an ammonium ion, would probably react with those, or like an ammonia NH3, right? But obviously ammonia cannot be there in the soil, so that's why we're saying NH4 plus. So ammonium ion, they would probably react with the organic compounds that form biomolecules in the plant cells. So that is why nitrogen itself is of no use, but nitrogen in a more reactive ion form, a mineral ion form, is of absolute use to the plants. So what the rhizobium does is basically it takes enzymes जो उसके पास है, उसके सेल के अंदर है, वो enzymes लेता है, nitrogen obviously comes from the air, and it makes those compounds, जैसे नाइट्रेट आयंस बनाया, अमोनियम कंपाउंड्स बनाया, और वो उसको इतना कंपाउंड तो चाहिएगा नहीं, जितना उसको चाहिए वो ले लेता है, बाकी जो एक्स्ट्रा बनाया जा रहा है वो इट काइंड ऑफ लाइक एक्सक्रीट्स दैट इनटू द सॉइल, बिकॉज वो उसके लिए एक्सेस है, और जो उसने सॉइल में डाल दिया है ना, जो उसने सॉइल में डाल दिया, अब वो सॉइल से प्लांट्स अपने रूट्स, जो लेग्यूमिनियस प्लांट्स हैं जो ग्रो कर रहे हैं, है ना, जो हमारे वेजिटेबल्स जैसे ग्रीन वेजिटेबल्स हम बोलते हैं, वो वेजिटेबल प्लांट्स दे आर जस्ट टेकिंग नाइट्रोजन फ्रॉम द सॉइल इन द फॉर्म ऑफ दीज रिएक्टिव मिनरल आयंस. तो वो लोग नाइट्रोजन बोल के नाइट्रोजन नहीं ले रहे, वो लोग नाइट्रेट आयंस, अमोनियम आयंस वैसे करके हमारे नाइट्रोजन को अक्वायर कर रहे हैं. और अब क्यों, अगर अगर हम नेचुरली नाइट्रोजन सप्लाई नहीं कर सकते, तो देन व्हाट डज द फार्मर डू, फार्मर पुट्स फर्टिलाइजर्स. फर्टिलाइजर्स क्या होते हैं, दे विल हैव लाइक अमोनियम कंपाउंड और अ नाइट्रेट कंपाउंड, तो उससे वो लोग को आर्टिफिशियली हमें नाइट्रोजन देना पड़ेगा. बट अगर नेचुरली उनको नाइट्रोजन मिल रहा है, तो ऑब्वियसली दैट्स लाइक बेटर एंड मोर चीपर फॉर द फार्मर एज वेल. बट बदले में राइजोबियम को क्या मिलता है? सो मोहम्मद, कैन यू टेल अस व्हाट advantage does rhizobium get when it, say, stays or inhabits close to the plant hosts?
Basically, the financial benefit, it reduce the
To the
cost of
rhizobium, to the
growing these.
rhizobium, like
Oh, to
it's a symbiosis, right?
rhizobium.
So how does the symbiosis work?
Basically, it depends on the plant for water and other sources to be used by the ribosome, and in
Absolutely.
exchange, uh, the ribosome give the plant nitrogenous compounds.
Absolutely. So we can see that, okay, the rhizobium will kind of like feed on the plant cells. We can see this image over here, right? It's just thriving over here and kind of taking nutrients from the plant xylems, from the plant phloems. So some carbohydrates and some nutrients that the rhizobium bacteria needs to survive, it will acquire them kind of parasitically through with the leguminous plants, but in return it will give them the nitrogen in the reactive forms that they will need. So that is what, that is how a symbiosis is established. Okay, now let's move forward with our next useful microbe, and that is yeast. Okay, so we all know what yeast is, है ना. हमने घर पे आपने ब्रेड बनाया होगा, आपने केक बनाई होगी, and if you have made those things, you have definitely worked with yeast. I mean, you might have not worked with them, but okay. So ना तो कम से कम होगा ही. Okay, so Mohammed, can you take us through the yeast cell?
Yeah, so basically, yeast cell is a microorganism. It is a fungi, and as you said, it is one of the most popular ones which we use for the making of bread. It is heterotrophic, which means it feeds on other organisms for the use of carbon compounds, and it reproduces asexually. It has a simple unicellular structure. It has a mitochondria, cell wall membrane, vacuole for storing fats. It has a bud scar, cytoplasm, nucleus, Golgi pouches, and lipid granule, which means basically it is a eukaryote. It is more developed than prokaryotes or bacteria because it has mitochondria and other small organelles which can make energy inside the cell. They don't need it from outside the cell.
बिल्कुल।
itself.
सीधी बात दोस्तों, यूकैरियोट मतलब न्यूक्लियस है, प्रोकैरियोट मतलब न्यूक्लियस नहीं है, राइट? प्रोकैरियोट्स डोंट हैव न्यूक्लियस, दे हैव लाइक अ डीएनए कॉइल, जैसे बैक्टीरिया डज नॉट हैव अ न्यूक्लियस बट इट विल हैव लाइक अ डीएनए इन अ सिंगल लूप ऑफ अ, यू नो, और like a coil basically. So that's how bacteria will have its DNA, and it will also not have a mitochondria. So that is the difference between a eukaryote and a prokaryote. Okay, now Mohammed, can you explain how this budding process works when there is this
Uh,
reproduction for yeast?
Reproduction.
Yeah,
So
so
basically,
yeah.
this is through mitosis, cell division. Uh, there is form phases. I don't think it is mentioned, but it is a little bit advanced, I think.
Okay, yeah, it is advanced, but okay, let's just go to simple fine. Okay, so like guys, एक काम करते हैं, मोहम्मद हमारे जो भाई है वो बहुत advanced है, right? He thinks like he's obviously studied way too much, so he won't be able to simplify it. Maybe I'll make them simple. आपको example से बताता हूँ, ठीक है? अब ये जो हमारा सेल है, राइट? ये जो जो सेल है, दिस सेल हैज़ प्रॉपर अपने ऑर्गेनल्स, प्रॉपर सब कुछ है. नाउ अचानक से the cells begins to like grow a protruding, you know, bud, जिसको हम बोल रहे हैं. आप ये इमेज में देख सकते हो कि एक adjacent bud यही सेल में ग्रो हो रहा है, राइट? माइटोसिस में क्या हो रहा है दोस्तों? Mitosis may be there is, just the chromosomes are splitting, they're duplicating and they're splitting in the kind of like the same cell. So it is very similar to mitosis, but इसमें एक अलग सा एक bud जैसा shape form होता है. There's this protruding bud shape, right, that is being formed, and एक एक करके all of the organelles will duplicate. So first a new bud will form slowly up, there is this circle over here first. Okay, let's just like kind of run through the process. So there's this circle. Okay, let's assume this is the nucleus. Okay, this is the first stage where there's a normal yeast cell. In the second stage, there'll be a bud that will start to grow. A bud will start to grow. Then on the third stage, a bud will be formed, and like a nucleus will kind of duplicate. Right, the nucleus will kind of duplicate. And finally, when all of the organelles have been duplicated, you can see over here the green organelles are duplicated, the white organelle is duplicated. When all of the cell organelles are duplicated, they are made, just copied. They are, you can say the makeup of those cells is and cell organelles is copied. Finally, we there will be two daughter cells formed. That means this cell will just split into two. This parent cell, it will just split into two. The cell membranes, new cell membranes will be formed that will now just separate this delineation, and two daughter cells over here will be formed, and this is just the process of budding. It's like similar to binary fission that happens in bacteria. It's similar to mitosis that happens in all other living organisms, multicellular ones. And but just it's just that there's this bud that grows out of
Yeah,
the,
it is a type of mitosis.
yeah, type of mitosis we can say. So that is what happens in budding. But okay, I also need to mention that guys, under like extremely stressful conditions, the you know the yeast can also reproduce sexually. So it generally reproduces asexually, but अगर बहुत ही like stressful conditions है, बहुत एक्सट्रीम कंडीशंस है, लाइक बहुत ज़्यादा पॉपुलेशन डेंसिटी हो रहा है, बर्ड ग्रो करने का स्पेस नहीं है ना, कंडीशंस फेवरेबल नहीं है इतने बर्डिंग के लिए, देन दे विल रिप्रोड्यूस सेक्सुअली एज वेल. ओके, so how does fermentation in yeast work, which is quite similar to fermentation in bacteria, but how does fermentation in yeast work?
So basically, yeast transpire anaerobically, but unlike the one which we studied, lactobacillium, I think, lactobacillium, here it doesn't produce lactic acid; it produces ethanol and carbon dioxide. So
Okay.
it has the same way because ethanol can be remade inside the cell, unlike glucose, which need oxygen. And because yeast respire anaerobically, so it has to make ethanol, but it also produces carbon dioxide, which can be seen when you ferment bread, for example. It will start to increase in size and form a bubble, which is filled with carbon dioxide, for example.
Wow. So that just means that the yeast cell is just casually respiring anaerobically, and that bubble is the carbon dioxide. Is that what you mean, Mohammed?
No, I mean that the yeast respire anaerobically under if oxygen is not available.
Okay. So, guys, there was one difference that the lactobacilli just preferred to, respire anaerobically. It naturally needed anaerobic conditions, right? It really cannot really survive in एरोबिक कंडीशंस, मतलब अगर बहुत ऑक्सीजन होगा तो वो उतना अच्छे से सर्वाइव नहीं कर पाएंगे. बट यीस्ट जो है, दे कैन सर्वाइव परफेक्टली वेल इन एरोबिक कंडीशंस. दे इफ देयर इज एम्पल ऑफ ऑक्सीजन, दे वोंट लाइक डाई, राइट? उल्टा वो लोग के लिए अच्छा ही है. बट in anaerobic conditions they can also reproduce, or they can also respire, right? So they can respire in both aerobic and in anaerobic conditions, but lactobacilli, they were really preferring anaerobic conditions. ओके, सो वैन अगर ऑक्सीजन हमने काट दिया सप्लाई, अगर उतना एयर नहीं है, जैसे दही है, वो दही के अंदर उतना ऑक्सीजन डिजॉल्व नहीं है, तो देन वो लोग फोर्स हो जाएंगे कि वो लोग को अब एनारोबिकली ही रेस्पायर करना पड़ेगा, उनके पास और कोई चॉइस नहीं है. एंड दैट्स हाउ दे रेस्पायर एनारोबिकली. एंड बिकॉज ऑफ दैट द शुगर इज ब्रोकन डाउन इनटू इथेनॉल एंड कार्बन डाइऑक्साइड. सो इफ यू जस्ट राइट द इक्वेशन फॉर दैट, जो हमारा ग्लूकोज या शुगर है, राइट, वो यहाँ पर हमारा and ethanol plus CO2 में break हो जाएगा. It is not completely broken down into carbon dioxide, right? This ethanol can further be combusted with oxygen and broken down either even further, right? But ये ethanol जो है, ये हमें कुछ कुछ uses में काम आता है, और ये कार्बन डाइऑक्साइड भी हमें कुछ कुछ uses में काम आता है, जो हम आगे अभी पढ़ेंगे. बट इट ऑल्सो रिलीजेज सम एनर्जी फॉर द यीस्ट सेल, है ना? यीस्ट को क्या फ़ायदा मिलेगा ये करके? यीस्ट क्यों कर रहा है यार एनारोबिक रेस्पिरेशन? बिकॉज उसको खुद को एनर्जी चाहिए उसके लाइफ प्रोसेसेस के लिए, और इसलिए वो उसको जो ये रिएक्शन है यहाँ प्लस एनर्जी भी मिलेगा यहाँ पे ना, एक्सोथर्मिक रिएक्शन है ये. So this reaction will give the yeast cells energy to survive and carry out its life processes. Okay, so that's what this fermentation process is in a nutshell. Okay, so how does this process help in bread making, Mohammed?
So basically, yeast use the glucose and the sugars present in the bread and the disaccharides present in the bread and break it down into simpler sugars to make basically bread by combining yeast, flour, sugar, and water and salt. We create an aerobic environment for the yeast, and the yeast started basically to feed on the flour, sugar, water, and salt, and produce a dough which is used to make a bread.
एब्सोल्युटली. सो देखो दोस्तों, सीधी बात है, ठीक है? हमने हमने क्या किया, हमने एक पतीला लिया, वी टुक अ बोल, एंड उसमें हमने जो ये इंग्रेडिएंट्स है, देखो, फ्लावर, शुगर, वीट क्या, वाटर, सॉल्ट एंड यीस्ट, है ना, ये सब हमने मिक्स करके एक आटा बना दिया, एक डो बना दिया. अब हमें शुगर क्यों चाहिए? बिकॉज जो ये यीस्ट है, ये यीस्ट विल फीड ऑन दिस शुगर, राइट? द यीस्ट विल फीड ऑन दिस शुगर बिकॉज दैट इज द सोर्स ऑफ एनर्जी. जो हमने यहाँ पर पढ़ा था कि फर्मेंटेशन के लिए हमको एक टाइप ऑफ शुगर मतलब हमारी चीनी नहीं, बट एनी टाइप ऑफ यू नो biological sugar like a glucose or a fructose or a disaccharide, polysaccharide, any type of sugar. So the yeast will feed on our sugar, and it will produce carbon dioxide and ethanol, है ना? तो हमारा जो फर्मेंटेशन होगा, इट विल कैरी आउट द फर्मेंटेशन रिएक्शन. अब आपने पढ़ा होगा, है ना, जब हम आटा गूंदते हैं, आटा गूंदने के बाद में ढक दो, है ना। जब आप अगर आपने ब्रेड बनाया होगा तो आटा गूंदने के बाद वो आटे को हम ढक देते हैं। तो अब दो चीज़ हो रही है, एक तो आटा ऑलरेडी वैसे टाइट है तो वो आटे के अंदर तो ऑक्सीजन ज़्यादा होगी नहीं, और हमने ऊपर से ढक दिया तो ऊपर से भी और ऑक्सीजन नहीं आ रही है। तो ये जो एक ऑलरेडी हमने डो बनाया और डो को कवर कर दिया, राइट, इट इज़ कवर्ड। तो ये जो कवरिंग का प्रोसेस है, दैट इज क्रिएटिंग एन एनारोबिक environment where there is not a lot of oxygen now inside this environment around the yeast when it is feeding on the sugar. So it is feeding on the sugar but it does not have a lot of oxygen. तो उसके पास अभी एक ही चॉइस बचेगा कि उसको एनारोबिकली रेस्पायर करना पड़ेगा। तो जब वो एनारोबिकली रेस्पायर करता है, हमें पता है कि जो रिएक्शन होगा, राइट, दैट विल प्रोड्यूस इथेनॉल एंड कार्बन डाइऑक्साइड। तो अब ये इथेनॉल का तो हमें कोई काम नहीं है, बट जो कार्बन डाइऑक्साइड है ये गैस है। अब जो मेरा आटा है, ये मेरा आटा है, ये आटा तो ऐसे एक प्रॉपर कट्टर सॉलिड है, कॉम्पैक्ट सॉलिड है, बट जो एक गैस है, अब एक गैस विल कॉज बबल्स टू फॉर्म इनसाइड द आटा, ये आटे में बबल बनेंगे, है ना। अब ये आटे में जो बबल बन रहे हैं, ये दिस बिकॉज दिस ऑब्वियसली दिस कार्बन डाइऑक्साइड इज गोइंग टू ट्राई टू एस्केप एंड दैट विल जस्ट कॉज माई डो टू राइज, है ना। सो इधर यू कैन सी द डो विल राइज बिकॉज ऑफ द एयर। दी हमें पता है एयर एक्सर्ट्स अ गैस प्रेशर, है ना, एंड ये जो बबल्स है दे आर एक्सर्टिंग अ प्रेशर ऑन द डो एंड जस्ट कॉजिंग इट टू एक्सपैंड एंड जस्ट अ लिटिल बिट ऑफ आई वुडंट से हॉलो, बट थोड़ा सा स्पॉन्जी हमारा डो बन जाता है, लिटिल सॉफ्ट डो बन जाता है। बिकॉज अब वो पहले वो कट्टर हार्ड एकदम कॉम्पैक्ट सब बंद था, बट अब वो हल्का सा एक एक्सपेंशन हमें दिखने को मिल रहा है बिकॉज ऑफ द कार्बन डाइऑक्साइड दैट इज जस्ट काइंड ऑफ लाइक इन्फ्लेटिंग the dough, I could say. Now, after that, Mohammed, can you explain us what do we do after the dough is risen? It's the step three. Could you explain us that? Explain
So
us step
basically,
three.
we have to keep it in a moist environment because it is more favorable for yeast. So
Absolutely.
it can reduce carbon dioxide, which is trapped in the dough, and make the dough inflate. So it will make it bigger, so it can be later be cooked into bread or cake or whatever you want to make from this dough.
Absolutely. And then after that, we just put it in the oven, bake it. Baking, what does it do? It kills the yeast and it will like evaporate the ethanol. Right. Another important thing that happens, it evaporates all the ethanol because ethanol is ultimately the alcohol.
Basically,
Yeah.
every time you eat a bread, you eat dead yeast with it also.
Yeah, you do. Right. So it just kills the yeast, it hardens all the outer surface. So if you just see the crux of it or the borders of the bread, the harder part of it, that's because it has been baked. And we can see spongy bread is prepared because over there, if you see, you'll see small little craters, small pores inside the bread, and that is because it has been you know the dough has risen due to the action of the yeast. तो अगर आप ब्रेड देखोगे कभी भी, और ब्रेड में हल्के हल्के आपको ऐसे खड्डे खड्डे दिखेंगे बीच में उसके, अगर आप स्ट्रक्चर में, तो वो इसलिए है क्योंकि जो यीस्ट था, वो यीस्ट ने वो डो को राइज किया और बीच में हल्का हल्का जो क्रिएट हॉलो इन्फ्लेशन यहाँ पर हुआ था डो के आटे के अंदर, है ना। अगर आप वो नहीं करोगे, जैसे कभी कभी अगर आप समझो आप रोटी बनाते हो, है ना, व्हेन यू मेकिंग फ्लैट ब्रेड, तो तभी वो इन्फ्लेट नहीं होता क्योंकि हमने यीस्ट ने डाला है ना। तो दैट एट दैट मोमेंट ऑफ टाइम अगर आप रोटी देखोगे तो रोटी के ऊपर भले ही वो कभी कभी वो खड्डे रहते हैं, बट दैट इज़ बिकॉज हमने उसको वो रोटी को सेका है, वो सेकने वाले वो कभी वो खड्डे रहते हैं, है ना। बट अगर आप रोटी का जो आटा है, रोटी आप तोड़ोगे तो उसमें आपको स्पॉन्जी स्पॉन्जी टेक्सचर नहीं मिलेगा। आपको वो रोटी का अगर जब आप तोड़ने जाओगे, आपको पूरा आटा 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 दिखेगा, आपको बीच में कभी भी वो तोड़ते वक्त खड्डे नहीं मिलेंगे। वो बैटर या यो जो रोटी के अंदर है ना, वो आपको नहीं मिलेगा बिकॉज वो डो जो था, जो आटा जो था, वो राइज नहीं हुआ है, वो अभी का अभी वो कट्टर का कट्टर आटा ही आपने उसको बनाकर सेका है वो रोटी को। एंड दैट्स व्हाट इज हैपनिंग व्हेन यू आर लाइक मेकिंग रोटीज एज अपोज टू making bread or baking cakes, right? So that is the process that is just taking place over here now. Antibiotics. Okay, we can finish antibiotics, Mohammed, today and probably take a break because it would just be a little too much to sink in.
Yeah, so basically, antibiotics is a chemical compounds which are used to kill and destroy bacteria cell wall to eliminate harmful bacteria from the body. The first discovered antibiotic was penicillin, which was discovered from a bacteria itself.
ऑसम सो देखो दोस्तों मोटा मोटी यहाँ पर मोहम्मद ने हमें बताया कि एंटीबायोटिक्स आर लाइक बैक्टीरिया को मारने वाले उपाय राइट व्हेनेवर वी से कि मैं बीमार हूँ आई एम इल यू नो आई हैव गॉट एन इन्फेक्शन so if it is a bacterial infection, if there's a bacteria, there's a harmful bacteria that is causing that disease, then antibiotics jab hum dawai lete hain, the pills that you get, those contain certain chemicals that kill the bacteria and they will cure the infection. So that is what is the work of antibiotics. Now you can read through this, but how do antibiotics actually work, Mohammed? Can you help us with the working of
So
antibiotics?
basically, they attack the cell wall of the bacteria and destroy it, which leak everything inside the bacteria outside the cell and diffuse it. So basically, all the organelles inside the bacteria become scattered across the place where it was destroyed.
Absolutely. So the antibiotics over there, they are also chemicals, and what they'll do is just they'll kind of, as Mohammed said, they will attack the cell wall and they will diffuse, or you can just kind of react with the cell wall. So the cell wall, जो hard structure जो है, it just it just gets converted into a different compound, and that reaction, say we can say, diffuses or makes it less harder, makes it kind of like a semi-solidish substance,
Hmm.
and the cell wall
And as we know
diffuses.
that
Yeah.
inside the bacteria is more concentrated, so it will diffuse outside the bacteria, and basically all the internal organelles of the bacteria will diffuse outside, and nothing will be left inside the bacteria.
Absolutely. So the internal organelles over there, they diffuse outside. मतलब basically तुमने अपनी जो नस है ना, वो काट ली. बेसिकली ये बात हो रही है कि जो अंदर जो भी ऑर्गेनल है वो सब बाहर आ गए. या नस नहीं काटी, अगर तुमने एक और मैं एग्जांपल तुम्हें देता हूँ, है ना. बिना अगर तुमने कोई सेल ऑर्गेनल काटे हुए, अगर तुमने मेरा जो स्किन है, अज्यूमिंग कि कुछ ब्लड लीक नहीं हुआ, तो भी तुमने अगर मेरा स्किन हटा दिया, सब all the, you expose all the organelles outside, then organelles जो है, they will start going here and there, because यहाँ पर कोई blood vessel नहीं है या nerves नहीं है जो उनको connect कर रही है ना. यहाँ अपने हमारे body में there are blood vessels that are connecting different organs, but over here everything is just randomly suspended in a solution, it is suspended in the cytoplasm. तो एक रैंडम पानी में एक रैंडम पानी का एक सोल्यूशन है, उसमें अलग अलग
Basically,
ऑर्गेनल्स पड़े हैं.
if
या
I want to simplify it, the bacteria is spilled all over the place and made into nothing.
बिल्कुल. तो वही हो रहा है, मतलब it's like there is this, the cell wall is like the security guard, and you're just killing off the security guard, the antibiotic is murdering the security guard, and you just now can go in and just loot up everything that's in there, right? That's what's happening. Or you can say that there's a door, and behind the door there is like a lot of treasure that's stacked to the brim, है ना. ऊपर तक खजाना भरा है या ऊपर तक पानी भरा है, और जैसे ही तुमने डोर खोला, सारा जो पानी है that overflows out of the room and into the corridors there, and that just everything that's dissolved or suspended inside that water also flows along with it, and nothing is left. So as Mohammed said, it just spills around, and that's how the bacteria just dies. So one such bacteria, that.
And I would like to add one last thing that after it spill out, the immune system will attack whatever is left out. So it will destroy everything and nothing will be left of the bacteria.
Oh, that's an important point, Mohammed, because, for example, there is this, we know that bacteria will obviously have enzymes that will attack
Hmm.
our cells and, cause diseases in us. And we'll also go through that in detail. So, if अगर enzyme ऐसे ही बाहर घूम रहे हैं, है ना, तो जो भी सेल मिलेगा उसको वहाँ पे अटैक करेगा, इट विल अटैक द नियर बाय सेल्स. सो टू डिस्ट्रॉय दोज एंजाइम्स, टू डिस्ट्रॉय दोज ऑर्गेनल्स ऑल्सो, our immune system, our WBCs, white blood cells, will attack those organelles and destroy those cells. So a very important point mentioned by Mohammed here. Now, well, the first antibiotic, as Mohammed said, was penicillin. And Mohammed, can you tell us a little about penicillin?
So basically, it was discovered by accident, and it is discovered from a bacteria that was being tested by a scientist, and which was left with another bacteria also, I think. And then these bacteria, one of these bacteria produced an antibiotic to kill the other bacteria and destroy it, which was later discovered by a scientist, which I cannot remember his name, but this was how it was discovered. So it treat all the bacterial diseases, not the viral diseases, because virus does not have a cell wall. So all the bacterial infections can be treated by penicillin or the antibiotics that have the same compound.
Oh, that's another interesting insight that Mohammed has shared, that antibiotics are not, efficient with viruses, because viruses do not have a cell wall. We will see the virus structure when we go to the harmful microbes and diseases, but viruses do not have a cell wall, and that is why it will not work on viral diseases, right? uh, so, and also the cell wall of bacteria is different in composition to a cell wall, say, to a fungi. Or if, for example, there is a plant that has been infected, right, and we are injecting antibiotics in the plant to resurrect the plant, then the plant will not be affected, because जो plant का cell wall है और जो bacteria का cell wall है, they are made up of two different, chemicals. So the antibiotic will only attack the chemical that makes the bacterial cell wall, and not other organisms cell wall. So that is also another important thing that you need to know. अब penicillin जो है, penicillin can treat diseases of ear, nose, throat, skin, syphilis, tonsillitis. These are some diseases that the penicillin bacteria can, treat, and we take penicillin quite a lot of times in our, you know lives. When we are ill, we take penicillin बहुत बार. Okay. Now, वन मोर थिंग दैट वी, डॉक्टर बोल डोज देता है, और हम बार बार वो रिपीट नहीं करते, हम बोलते हैं ओवर डोज नहीं करना चाहिए. सो मोहम्मद, कैन यू टेल अस व्हाई वी शुड, take antibiotics only when it's needed, and only in a particular limit?
So basically, you should never take antibiotics unless it is prescribed by your doctor to you, because using any antibiotic without any previous knowledge will might not take it is effect on the bacteria. Also, overusing of this antibiotic might allow the bacteria to develop resistance and mutate and make a more stronger bacteria which are resistant to this antibiotic, which now will force the scientists to find another way to kill the bacteria by developing another antibiotic, which is a lot of waste of time and money also. So antibiotics should be used only if it is prescribed by the doctor and within a certain doses and limits,
Wow.
so that, as I said, the bacteria doesn't develop resistance and making basically the antibiotic useless for the bacteria.
Okay. So you spoke about antibiotic resistance. We'll come to that, but there's also this point of completing the dosage of a, you know antibiotic when it's prescribed. Like, when we say that ठीक है 2 दिन में ठीक हो गया, in 2 days I feel I'm better and now I'm
Yeah,
cured.
yeah, you have to use the complete prescribed dose by the doctor. You should never use a small dose of it because you felt better the next day or something. Otherwise, you will feel better and the bacteria might go away, but you might develop a resistant type of bacteria inside you, which will be transferred to other people who will have these uh, develop mutated resistant bacteria inside them, and they cannot use the antibiotics that you used because you felt that I felt better the next day, so I don't want to use the full dose. I will use just little bit that is
Absolutely.
making me feel good, and that's it.
Absolutely. And guys, see, if you're feeling sick, that's because it's not because one bacteria destroyed one cell. Right? It's because like say hundreds of bacteria just like disturbed like hundreds of cells, and that is why your body is feeling sick. So if you're feeling better now, if you think you're cured, that means like most of the bacteria is killed, but you still need to kill all of the bacteria cells, because even if like one bacteria is left, even if one bacterial cell is left, they can really reproduce quickly and multiply again. So it's important to kill all the bacteria and not even leave a single strand of bacteria. So that's again another reason why you should complete your dose. Now, Mohammed, last thing before we bid goodbye: antibiotic
Hmm.
resistance. How does this work? Because there is this DNA, right? You spoke about mutation. So if I just
Hmm.
make up this bacterial cell over here, okay, and pardon my drawing, it's horrible. So we have the coil of DNA over here. And can you explain what mutation means first?
So basically, every living organism contains genetic material which we call DNA. This DNA has a sequence of letters, and each sequence corresponds for a protein, an enzyme, an organelle inside the bacteria inside the cell, which will make this whatever uh, compound that is needed by the cell. Basically, everything that you need is prescribed in this DNA, and each compound has a sequence, specific sequence. Now, when a mutation happen, it is because the bacteria has made a specific sequence for a specific enzyme which can break down the antibiotic that you just overused or you used with lesser amount than the prescribed dose. So it allows the bacteria, it gives the bacteria time to mutate and develop resistance or develop a code which make an enzyme which can break down the antibiotic and basically make it useless and ineffective.
Absolutely. So it means that abhi dekho, mutation can happen for random reasons. So but there's a very little chance that it happens. But if there are, for example, a million bacteria in your body, there is a
Hmm.
possibility that one of them can mutate and one of them can develop the antibiotic resistance. And that means that if they detect the antibiotic coming close to your cell wall, they will release their enzymes, they will release their you know solutions that will go and negate the antibiotics. So they will react with the antibiotics and just kind of destroy the antibiotics. So bacteria will release some enzymes of its own that will react and break down the antibiotics. That means that antibiotic सेल वॉल तक पहुंचेगा ही नहीं. मतलब क्या हो गया कि अगर पता चल गया, security guard just got that feeling that there are people who are trying to infiltrate, there are people who are attacking, so it just wakes up like a few more people who are sitting behind, or you could say it just calls for reinforcements. It calls up the police and say the police comes and stops the burglary. So that is kind of what's happening. The reinforcements are made and all the thieves are negated even before they can reach the security guard. तो सिक्योरिटी गार्ड के पहुंचने तक ही उनको रास्ते में ही मार दिया जाता है. पुलिस आती है, पुलिस रास्ते में ही उनको मार देती है ताकि वो सिक्योरिटी गार्ड तक पहुंच ही नहीं पाता. सो दैट इज व्हाट इज हैपनिंग. पर
So
उसकी
that's why you
या
have to complete your dose, because when you use a smaller amount of dose, it will leave some bacteria behind, and it will give this bacteria time to discover the antibiotic and develop and mutate to produce an enzyme which will break down this antibiotic. Basically, you are exposing the bacteria to small dosage because you didn't use the whole prescribed dose.
absolutely right. Now how does this work and why does this antibiotic resistance develop? There's a specific chapter in heredity and evolution in grade 10 that we will study about natural selection and survival of the fittest. ये हम पूरा पढ़ेंगे, but that's not for this chapter, so we won't go very deep into that. But you can just understand that one of or the, one of the bacteria can mutate and it can develop antibiotic resistance. So to and then जितना भी दवाई खाओ कुछ फर्क नहीं पड़ेगा. So to prevent that, please take your dosage, complete your dosage, but at the same time don't overuse a particular antibiotic. Also, you can try using a combination of antibiotics so that the bacteria is not habituated to one type of antibiotic. You know, so there are different ways you can mix and match, and that is why you should take only the prescriptions from your doctor. Your doctor knows your history. Your doctor knows कि ठीक है क्या क्या mix and match में करके दे सकता हूँ. What combinations will be effective, and only take the antibiotics that your doctor prescribes for you. So these were some very useful microbes that we learned about. We learned about lactobacilli, rhizobium, and yeast. We also spoke about antibiotics and how they can be used to kill bacteria. But in the next lecture, we will speak about the harmful microorganisms. That is the second half of this chapter, and we'll also be solving some summary questions, some end of exam style questions that will simulate कि कैसा होता है एग्जाम में. और जैसा हमने पहले भी डिस्कस किया है कि यू नो हमें समझना एक बात होती है और एग्ज़ाम में वो प्रेजेंट करना एक अलग बात होती है. तो वो कैसे ये चैप्टर को हमें एग्ज़ाम में प्रेजेंट करना है, किस पॉइंट्स क्या क्या इंपॉर्टेंट पॉइंट्स हैं, और हम एग्ज़ाम जो आंसर्स लिखते हैं वो कैसे स्ट्रक्चर कर सकते हैं, वो हम डिस्कस करेंगे अगले लेक्चर में. Can you hear us now, Mayur? Hello, are we audible now? So that is what is. Hello, hello. Can you hear us now? Yeah, I think we're fine. Okay, sorry, Mohammed. We were talking about Mohammed's passion for this subject, and I just said that he's really passionate about biology, microbiology, and
Yes.
biochemistry, and he's really working on a research project with one very great professor. I don't know if the professor would like me to divulge his name out right now, but Mohammed, can you share some details about your experiment?
So it is a research on Staphylococcus bacteria. So this bacteria basically uh, a bacteria that exists on the skin, and it is very harmful if it gets inside the human body. So we are studying how does the bacteria develop resistance if we expose it to small amount of herbal antibiotics, and basically we are the specific amounts, small doses, and we increase the doses each time to allow the bacteria to develop resistance. And just like how we explained now, we expect the bacteria to develop resistant to the antibiotic. So basically, the antibiotic will become useless when it is applied on the bacteria. So this is what is the experiment about. It is the resistance of of the bacteria Staphylococcus, which
So
exists on the
yeah.
behind the ear, on mostly on the skin, and inside on the nose also.
So, like Mohammed is, I think, really passionate to save the world, help the world be a better place, and these researches just helping people to understand more about. He's obviously he's simplified the experiment for us. I'm sure it wouldn't be, as easy as it sounds, and he said that it's quite hazard working with that bacteria, as it can cause lethal problems to the skin. So I'm sure he must require taking great care, great caution, and great risk as well. So thank you to brave hearts like Mohammed, who are just expanding the field of STEM, of science, and really helping us
Thank you
grow.
so
And thank
much.
you so much for Mohammed for taking time out at this hour and coming to help us with our syllabus, our curriculum. So thank you so much, Mohammed.
Thank you for inviting me, and it is my pleasure. Um, you can always rely on me when it comes to these topics.
Yeah, and I think Mohammed is the best man indeed for these topics. So, guys, if you have any questions for Mohammed for the stream, please put them on in the comments, and I'll try to reply. And if I can't, I'll fetch Mohammed out from wherever he is in the world, and he may be working on some groundbreaking research, but he will still. I will make sure that I get a grade 9 answer from him, and reply to you at the soonest. So that's it from our side today, guys. If you like the stream, please subscribe to the channel and like the stream as well, and that gives us really more motivation to make such quality content for y'all. We'll see you guys in the next lecture. I discussed about the plan for the next lecture that would be studying about the harmful microbes and also revising the exam style questions, because knowing the concept is one part of it and presenting it in an exam is another. तो हम वो वाला एस्पेक्ट भी डिस्कस करेंगे कि ये चैप्टर में क्या की पॉइंट्स है जो हम एग्जाम में हमें लिखना है और कैसे है वो एग्जाम के आंसर्स को स्ट्रक्चर करना है. So we'll see you guys next time. Mohammed, any last words would you would like to say before we leave?
I think we explained everything clearly. Uh, so yeah, that's it for today. Again, thank you so much for inviting me. It is my pleasure to explain uh, biology for younger students and clarify doubts for them.
Thank you so much, guys. We will see you guys tomorrow or in the next lecture. Updates के लिए Instagram follow कर दो. तब तक के लिए bye bye. Thank you so much.